Hello and welcome back to the another series that is molecular biology. So till now we have been discussing about what are vertebrates, what are invertebrates, what are the evolution, etc., etc. But in this video series we are going to deal with the molecular biology. What is molecular biology? These are the molecules that is mainly dominant our life, our all the functions of our body, that is mainly the DNA, how the DNA, RNA, what are the roles of their, we will be discussing in this series. So for the first part of the series is the structure of the DNA. So we will be discussing what is the structure of the DNA. So let's start without any delay. And if you have not subscribed to my channel, before that do subscribe, like and comment. Let's start. Before moving, what is the structure of DNA? Let's know what is the nucleic acid is all about. So nucleic acid is a biomolecule, you can say, which was first discovered by Frederick Mischer, is the name of the scientist. And nucleosin is a molecule which is a mixture of basic protein and phosphorus containing organic acid. So by the mixture of these protein and phosphoric acid, they form the organic, that is the nucleic acid. That's why they are called nucleic acid. So you can say nucleic acid consists of protein, okay, protein, and what else? Phosphorus containing, one is phosphorus containing nucleic acid, that is known as the what? Nucleic acid. Okay, so nucleic acid is of two type, that is ribonucleic acid, that is known as RNA, and deoxyribonucleic acid, that is known as the DNA. Why so? Both are nucleic acid. What is why the RNA is called ribo and why DNA is called deoxy? Because of the sugar molecule. We will be discussing later on. So this is the basic structure of the DNA and this is the RNA. So as you have known that the DNA is a double helical structure or is a double stranded structure and basic genetic material for all the life organism, and some viruses also contain the DNA as the genetic material. But RNA also sometimes can act as a genetic material, which is usually a single stranded structure, you can see, and these are also acts as a genetic material for some of the virus. When we will be discussing about the what is the difference between DNA and RNA, after we discuss about the RNA structure, we will be comparatively dealing what is the difference between DNA and RNA. So for now we will be discussing about the nucleic acid. Next, moving to the nucleic acids are mainly known as the monomeric unit, or you can say each nucleic acid is form of the nucleotide. Certain units will be formed, that is monomeric means single unit. Okay, the single unit is known as the nucleotide. So what are the nucleotide consist of? What is the nucleotide is formed? Nucleotide consists of three components, that is a base that have nitrogen atom, we can say nitrogenous bases, a 5 carbon sugar and a phosphoric acid. So these are the three components, major components of the nucleotide, and that's why the nucleic acid is called as the polynucleic acid. What is poly? Poly means many. So nucleotides is the mixture or the many nucleotide forms the nucleic acid. And what are the components of the nucleic acid? That is the base, nitrogenous base, which will be dealing later on, carbon sugar and phosphoric acid. These are the three components of the nucleotide. First we will be dealing with the nitrogenous bases. What are nitrogenous bases? Nitrogenous bases are the heterocyclic planar. What do you mean by hetero? The two different molecules will be present, and cyclic because they are present in a cyclic manner. Okay, you can see the structure. The major two nitrogenous bases are purine and pyrimidine. What are purine and what are pyrimidine? We will be dealing now. Okay, first dealing with the purine. As you can see the structure, this is the general structure of the purine. So the general structure of the purine consists of a 6 carbon compound, that is a benzene ring, with a 5 carbon compound and a nitrogen attached to it. So this is the basic structure of the purine. Moving to the pyrimidine. Pyrimidine consists of only a single 6-member carbon ring-like structure, and nitrogen is also present. So that's why they are called as the nitrogenous bases. As nitrogen is present, that's why they are called as the nitrogenous bases. First we will be dealing with the purine. So purine are of two types. One is adenine, another one is guanine. So in the structure itself you can see that as two rings will be present. That's why so purine. Remember that purine has two ring-like structures and pyrimidine have only single ring-like structure. So don't get confused with that. And purine category belongs to the adenine and guanine, okay, and pyrimidine consists of the thiamine and cytosine. In case of the DNA, mark that in case of the DNA only thiamine and cytosine will be present. But in case of RNA, instead of thiamine, what will be present? Uracil will be present. This is the difference between the structure of DNA and RNA. So, purine, that is the adenine and the guanine, adenine has an amino group in the C6 position. You can see that is the amino group in the C6 position, okay, and NH2 group in the C6, this is amino group in the C6 position. Let me erase it. Okay, so amino group in the C6 position, and guanine has amino in the C2 position. You can see this is the second position, amino group, and carbonyl at the C6 position. So this is the carbonyl, C double bond O, carbonyl is present in the C6 position, and C2 position the amino will be present. So position of the amino group is very, very important. Moving to the pyrimidine, pyrimidine are of two types, that is cytosine and thiamine. In case of RNA, uracil will be present. So they are single ring like structure, benzene ring like structure, and methyl group will be present at the 5' position of the thiamine. You can see CH3, that is the methyl, at the 5' position methyl group will be present in case of the thiamine, and cytosine the amino group will be, that is NH2 group will be present at the 4th position, okay. So and uracil is present mainly in case of the RNA. When we will be dealing with the RNA structure, we will be reading about the uracil. So these are the structures of the nitrogenous bases which we were dealing with. Moving to the carbon sugar, as we have discussed the first part of the nucleotide, that is a nitrogenous base. Second is the carbon sugar. So the carbon sugar is mainly, that is in case of DNA, deoxyribose sugar is present. That's why the name of the DNA is deoxyribose, okay, and RNA is ribose, because deoxyribose is present. That's why the DNA is called deoxyribonucleic acid. So deoxyribose sugar will be present. What is the deoxyribose? It is a 5-membered ring structure which will be lacking oxygen. Oxygen is mainly absent in case of deoxyribose sugar at the 2' position. This is the 2' position, is the first prime. This is the numbering: first prime, second prime, third prime, fourth prime, and fifth prime. So in the second prime position, the oxygen will be absent in case of the deoxyribose. Instead of that, what will be present? A hydrogen atom will be present. But if you see the RNA is ribose sugar, that is which is present in the RNA, OH is present. So this is the major difference between the carbon sugar of RNA and the DNA. That is, OH will be present in RNA, and in case of the DNA, H will be present. Next, moving to the phosphoric acid. So phosphoric acid is the major element of the DNA. What is this function? It acts as a glue. Okay, it acts as a glue, and this it mainly react with the sugar molecule and forms the ester bond. So where it is added? It is added at the fifth position, okay, of the nucleoside. It is added to the 5th position. So this is the structure of the nucleoside. And what is nucleoside? So nucleoside is a combination of sugar and nitrogenous base. That is, if phosphate is absent, only the sugar and nitrogenous base will be present, it is called nucleoside. And if phosphate is present, it is called nucleotide. This is the difference. And the bond between the nucleus, that is sugar and nitrogenous, the bond is mainly what type of bond? N-glycosidic bond will be present. Okay, N-glycosidic bond will be present. So this is the difference between the two structures. So this is the structure of the nucleoside. This is the sugar molecule, and this is the, what is the nitrogenous base. If two ring will be present, that is purine, and by this N-glycosidic bond they are attached. And this is the, if pyrimidine will be present, then single ring one. So this is the structure of the nucleoside. Next, moving to the functions of the nucleotide. What are the function of the nucleotide? We have discussed the three parts of nucleotide. What are they? First is the nitrogenous base, carbon sugar, and phosphoric acid. These three forms the nucleotide. Then what are the function of the nucleotide? They are mainly ATP currency, and they are the precursor of certain coenzyme like the NAD, NAD+, and also the polynucleotide helps in the transmitting of the genetic material. This is the structure of the nucleotide. This is the sugar molecule. Okay, this is the sugar molecule, that is deoxyribose. This is the phosphate group. Okay, this is the phosphate group, and this is the nitrogenous base. So these three, total three forms the nucleotide. Hope you have been clear with this topic. Next, moving to the structure of DNA. So the DNA structure was first described by Watson and Crick. Two scientists, they proposed the structure of the DNA. Okay. Next, mainly Watson and Crick described about the B form of DNA. What are the different forms of DNA? We will be discussing in the next video. But what are the characteristics of the B form of DNA? First, let's discuss that. Okay. First is that two polynucleotide chain. This is one polynucleotide chain. Okay. And this is another polynucleotide chain will be twisted around each other. So that's why they are two helical ring or two helical strands are coiled around each other. These are right-handed helix structure, that is they are turned towards the right hand side, and one strand moves from the 5 prime. If this strand moves from the 5 prime to the 3 prime direction, okay, if one strand moves from the 5 prime to 3 prime, another strand will be moving from the 3 prime to 5 prime direction. Okay, another strand will be moving from the 3 prime to 5 prime. So that's why both the strands of the DNA are located anti-parallel to each other. And one more important thing is that they are attached by the nitrogenous bases in between them. So the nitrogenous base, phosphoric acid, and carbon sugar forms the ladder form. Okay, they are joined by what type of bond? That is hydrogen bond. So hydrogen bonds are present between the base pairs. This ladder, like you are seeing, these strands are made up of nitrogenous bases. Okay, so these are nitrogenous base and phosphorus alternatively will be present. And these bonds, what are the type of bond will be present? Is the hydrogen bond between the base pair. And one more important thing to be noted, guanine binds with that of the cytosine, and important rule is that one pyrimidine, one purine will forms the base pair with one pyrimidine. Okay, so this is the rule. This is the rule. So guanine forms the bond with that of the cytosine. They have 3 hydrogen bond between them, and adenine forms 2 hydrogen bond with that of the thiamine. Okay, so this is the rule which is mainly known as the nitrogen base pair rule, which was proposed by Chargaff. Okay, Chargaff is a scientist who proposed this rule. And due to the interaction between the bases, you can see that is minor groove and major groove will be formed. That is a hole will be formed. Okay, due to interaction, you can see here what is minor groove and what are major groove. Due to the interaction between the base pair, that's why the minor groove and the major groove will be present. And this is the length of the base, that is the hole structure, and this is the diameter 20 Armstrong, and between each strand there is a gap of 3.32 Armstrong. Okay, so this is all about the structure of DNA, and this is a Chargaff rule that I have already told you, that the adenine and guanine that is guanine is unit is equal to cytosine and adenine is equal to the thymine. That's why guanine is forms the bond with that of cytosine and thymine forms the bond with that of the adenine. And one more important rule is that the DNA varies from species to species. Okay, so the DNA structure, that is the composition of the DNA, will be varying from species to species. This is the important point to be noted down. Okay, one second. Okay, this is the important point to be noted down. So this is all about the structure of the DNA, which I have described in detail. What are nucleotide, what are nitrogenous base, what is carbon sugar, what is phosphoric acid, all them I have detailed. But in the next class we will be dealing with certain other properties of DNA, that is what is supercoiling, what is coiling, what are the different forms of DNA, as well as what is denaturation of DNA. We will be discussing in the next video. Hope you have been understood this, what is the structure of DNA. If you have not subscribed my channel, do subscribe it, like, and maximum share among your friends, because these are very basic and important for exam point of view. Thank you and have a nice day.